We're going to be reviewing DNA and RNA and proteins, and I don't go into that much detail on this. I have no doubt, especially those of you who have had majors bio, those of you who might have even had a really strong Bio 3 teacher or microbiology teacher probably went into this in more detail than I'm going to. But we just want to make sure that everybody has a common understanding of this. All right, so I begin by reminding you that there are 46, and I'm going to use the term chromosomes, in the nucleus of every somatic cell. What's a somatic cell? A body cell. Soma means body. All of our body cells are genetically identical. All of our body cells came from the very first cell we started out with as a zygote. You remember that a sperm has the haploid number of 23 chromosomes. So does the ovum, the egg, with the haploid number of 23 chromosomes. When that sperm united with the egg, it formed the first body cell, commonly called a zygote. Commonly called a zygote, that's the first body cell with 46 chromosomes, or another way of saying that is 23 matching pairs. Because for every chromosome in the sperm, there was one that matched it as far as carrying similar types of genetic information in the egg. So 23 matching pairs. Here we wrote the zygote is the first body or somatic cell. Every body cell or somatic cell has a copy of these original 23 pairs of chromosomes. All right, now we're going to remind you of the chromosomes, and we're also, I wrote right here, each chromosome itself is made up of or contains about 2,000 genes, 2,000 genetic instructions. Let's look on the next page, page B14. And on page B14, what we're looking at on the top is commonly known as a karyotype. What does karyo mean? Anybody? Karyo. Now, all my students who've had anatomy from me should know this. Karyo means nucleus. You remember this? Prokaryotic cells, eukaryotic cells, right? Karyokinesis, karyotype. Karyo means nucleus. Prokaryotic and eukaryotic cells refers to those cells that lack a nucleus or have a nucleus. So it's always good to learn these roots. So a karyotype is when we analyze what's in the nucleus, which are these chromosomes. It's a chromosome analysis. So we had just said that you have 23 pairs of chromosomes. You had that in the zygote, your first body cell, and in every other body cell that develops by mitosis from it. Here we see of the 23 pairs of chromosomes, there are 22 pairs of autosomes, and 1 pair of sex chromosomes. The autosomes carry genetic information about genetic information such as pertaining to eye color and blood type. The sex chromosomes obviously contain information, not only this, but it carry information that in part determine your gender or what sex you are. Of course, if you have 2 large X-sex chromosomes, you're a genetic female. If you have 1 large X and 1 itty-bitty Y sex chromosome, you're a genetic male. All right, so those are the chromosomes. Now, if we look on page B15, so on B15, it says human chromosomes. Now, this shows pictures of the chromosomes, of the autosomes and sex chromosomes, diagrammatically, schematically. These are not what they actually look like, but it represents them. Each of these lines are genes. Those are genes. All right, so all of these different lines represent genes that are on these chromosomes. How many genes are there typically on each chromosome? About 2,000. We just said that 2 pages ago, about 2,000 genes. So, and so here it has a picture of autosome number 1, autosome number 2, autosome number 3, and 4, and 5, and 6. In the next row, autosome 7, 8, 9, 10, 11, 12. In the third row, autosomes 13, 14, 15, 16, 17, 18. And on the bottom row, autosomes 19, 20, 21, and 22. In reality, in the nucleus of every body cell, you have not one of each of these, but two, a pair of them. You have 22 matching pairs of autosomes. If we trace them back where they came from, 22 of them came from the egg from your mother, 22 came from the sperm from your father. Now, over here, we have the sex chromosomes. If you're a female, you have two of these large XX chromosomes. If you're a male, you've got one large X and one itty-bitty Y. Where did they come from? Well, if you're a female, one of the X's obviously came from the sperm from your father, and the other came from the egg from your mother. If you're XY, which one came from the mother and which one came from the father if you're XY? X from the mother and the Y from the father. The only one who can contribute a Y is the male, so is the father. So, because guys are XY. All right, so what these lines indicate are genes, and what are genes? So I think we wrote at the top, genes are the instructions for making proteins. They are the instructions for making proteins. It's important you understand what genes are basically for. Now, I'm going to walk you through some examples of these genes and the proteins they code for. And we're going to be talking about the majority of all these proteins in this course. I just circled some of these. We're not going to talk about all of them right now. Right here, there's a gene on autosome 1. I'm not asking you to know which chromosome a gene is on. I promise not to ask you that. But I want you to know what these genes are for. There is a gene on autosome 1 that has the instructions for making a protein called amylase. That's what genes are, the instructions for making a protein. What kind of protein is amylase? Okay, if it's just by the fact that it's called amylase, it's an enzyme, right? Because we know that almost all enzymes, the way we name them, are endinase. And in fact, we've learned that arguably the most important proteins in a living thing act as enzymes that catalyze chemical reactions. What does amylase do? Anybody know? Good, excellent. It breaks down starch. Remember, the technical name for starch is amylose, right? We learned that back in section A. So this is a digestive enzyme that allows us to break apart starch in our food. Now, if somebody has a defect in this gene, and really to be entirely complete, a defect in both autosomes that have, you have a pair of autosome number 1s. If they have a defect in that gene on both of the pair of autosome number 1, then they cannot make this digestive enzyme. That's a genetic disease, a genetic defect. Now, you don't die because you can't make amylase, but it certainly makes life difficult. Because if you can't digest starch, you can't eat any food containing starch. And that's everybody's favorite foods, as we talked about last class meeting. So when there's defects in these genes and somebody can't make a particular protein, it might be not serious at all, it might be pretty serious, or it might be fatal. Depends on exactly what the function of that protein is. Just to look at some other examples, you'll notice also on autosome 2, and I'm not asking you to know which autosome, there is a gene for making antibodies. Antibodies are proteins. We had reviewed that previously. Now, if somebody has defects in the genes for making antibodies, that's probably going to be fatal, because antibodies are what defend us against the bad guys, like bacteria and viruses. And if somebody can't produce antibodies against the bad guys, I think it's over. Also, there's a gene here for making a protein hormone called glucagon. Didn't we already learn that both glucagon and insulin are protein hormones? So the instruction for making glucagon is on this autosome. On autosome 3, there's a gene for making a protein called rhodopsin. Anybody know anything about rhodopsin, that protein? Huh? Anybody have any idea what it's for? Vision. Very good. Jessica's got it. So that's a protein. We're going to talk more about the role in vision, in seeing. when we get to visual physiology. We'll get into physiology of vision. So if you have a defect in that protein, then in that gene, so you're not able to make rhodopsin, it's going to greatly impair your ability to see. We're not going to go through all these. You'll notice that here is a gene labeled MHC. And we've mentioned, actually we haven't mentioned MHC, but we will be talking about MHC. Some of you might have learned it in your other classes. MHC stands for Major Histocompatibility Complex protein. You certainly learned that. If you had me for anatomy, you learned it, but you may have forgotten it. If you had microbiology, you certainly would have learned this. You might recall vaguely, if you've had micro, something called MHC type 1 or class 1 and MHC class 2. What are major histocompatibility complex proteins? Anybody know? Some of you have micro. You should definitely know that if you have micro. Those are also called recognition sites or cell identity markers. They allow your white blood cells, your immune system, to recognize which cells belong to you and which cells are foreign. So that way, when a bacteria enters your body, your white blood cell will attack the bacteria and not attack your own cells, because your cells have these identity markers. And they're called histocompatibility because it means that makes your cells compatible, tissue compatibility. We'll have more to say about this. On the next row, just to mention a few proteins, you'll notice here on autosome 7, again, you don't have to know which autosome, there's a gene for making a protein called collagen. Did we talk about collagen previously? In this class? Yeah, so where's collagen found in the body? Okay, good. So connective tissues, it's in the dermis of the skin. Tendons are high in collagen. You'll recall we reviewed how fibroblast cells and other connective tissue cells manufacture this protein called collagen. Anybody remember what vitamin is necessary to make it? Vitamin C, ascorbic acid. So we learned about that. So we're trying to integrate all this information. Now I understand the obvious response is, gosh, there's so much to remember. It's hard to remember. I understand. But as we remind you, when you eventually are a nurse, a PA, a pharmacist, whatever you're going to be doing, you're supposed to have that information at your command. Right? It's not like you say, you have to build up all this data bank so you can actually treat their problem and explain what their problem is. You can't just say, I had anatomy and I forgot it. I had micro, I finished it, I don't remember it. Okay? Because if you keep forgetting everything you learned in each class, how are you going to put it all together and treat a sick patient? It's a challenge, but you can work on it. Just keep studying hard. All right, on autosome number 9 are genes that determine what blood type you are. And when we talk about you being blood type A or blood type B or blood type O, what we really mean are the proteins that are on your red blood cells. Those are proteins on the surface of your red blood cells. And so if you're blood type A, you have what are called A proteins. And if you are blood type B, you have B proteins. So that's what genes are for, is making these proteins. On autosome 11, it's very interesting. You have the gene that has the instruction for making hemoglobin. We've learned about hemoglobin. Hemoglobin is the red-colored protein that transports oxygen in a red blood cell. Now, what if somebody had defects in their genes for hemoglobin? What disease would that result in? Sickle cell anemia. That's what sickle cell anemia is. Defects in that gene, and the person cannot make normal hemoglobin. So it causes the whole red blood cell to be misshapen. They call it sickle-shaped. And it doesn't, the hemoglobin's wrong, it's defective, it doesn't work right, it doesn't carry oxygen well. That's why it's called sickle cell anemia. They're anemic. Anemia means you can't carry enough oxygen. That's what anemic means. So genetic diseases result from defects in these genes. They result from an inability to make proteins. Also on that very same autosome is a gene for making the protein hormone insulin. So if somebody has defects in that gene, what disease is that going to result in? Diabetes. Again, this is not a complete listing by any means of all the genes that are on each of the chromosomes, just a few examples. There's also a gene on that very same autosome for making a brown-colored protein called melanin. Melanin is what creates pigmentation in our body, in our skin, to protect us against harmful ultraviolet rays. And that pigment melanin also appears in our hair and in our iris of our eyes. If somebody has a defect in that gene, they can't make that brown-colored protein. So they have a disease, a genetic disease called albinism. They're an albino. Totally white skin, white hair, and pink eyes. They have no pigmentation. Now, you don't die from being an albino, but you've got to be really careful of ultraviolet rays from the sun. You have no protection at all. So again, some of these genetic diseases, when you have defects in these genes, are not too big of a deal. Some are somewhat of a deal, and some are fatal. They're very serious. Now, in the third row, just to mention some others, there's a gene on autosome 13 called the BRCA gene. Has anybody ever heard of that? Anybody know anything about it? Okay, I'm assuming people are shy. Okay, I'm assuming most of you do know this. The BRCA gene, if there's a defect in this BRCA gene, it greatly increases the risk that a woman will develop breast cancer. A gene that can, a gene where a defect in it can result in an increased risk of cancer is known as an oncogene. Because what do we call the department that deals with cancer in a hospital? Oncology. That's a cancer gene. So they can now test for a lot of these genes. And so what's actually happening, I'm sure you've heard stories of this, you've heard of young women who make a very difficult decision, but they have radical mastectomies performed on both breasts at a young age because they know they've inherited this defective form of the gene, and it's only a matter of time before they develop breast cancer. And they may have a family history where their mother died from breast cancer, maybe an older sister died, an aunt died, and they know they're next. And they can test to see if they've got that defective gene.
So when a man gets breast cancer, it's like really rare, but it happens?
It does happen, and it's also related to the BRCA gene.
So he gets it from his mom?
Remember, you have two of these. Of each of these autosomes, you have a pair of them. And one of them came from your mother and one came from your father. So it could come from either side. But obviously, because of the nature of breast tissue and so on, and the hormones that affect breast development, it's more of an issue with women. Also, another example of an oncogene is on autosome 17, the p53 gene. Anybody ever heard of that one? Anybody know what a defect in the p53 gene results in? A defect in the p53 gene greatly increases the risk of colon cancer. What's the colon? What's that? The large intestine. And in fact, colon cancer is the second leading cause of death from cancer. Anybody know what the number one type of cancer is that causes death? Lung cancer. All right? But the number two cause of death is colon cancer. Now, the most common cancer is neither one of them. The most common cancer is, anybody know? Skin cancer. But skin cancer, because it's right on the skin and you see something's off, it doesn't look right, they catch it early. Right? But usually in terms of lung cancer and colon cancer, by the time somebody's feeling pain from these parts of the body where the cancer is growing, it's usually metastasized too extensively to save them. All right, again, we have tests now for these genes. You'll notice here's a gene, it's labeled Tay-Sachs disease. That's actually what that Tay-Sachs disease gene is. It's actually a gene, I'm not asking you to know it, I'm not. It's a gene for making an enzyme in the brain called hexosamidase A. Hexosamidase, it's all one word, amidase A. And you don't have to know it anyhow. So I can't even read it. Okay. Hexose, which means like six carbon sugar, hexose, amidase A. And a defect in that gene causes a deterioration of the brain. And a child born with Tay-Sachs with a defect in that gene is only going to live 2 or 3 years, and they die. No treatment. Again, all kinds of genes. Here's a gene for making a protein called myosin. Right? That's a protein in muscle cells, actin and myosin. And growth hormone. Did we ever mention growth hormone is a protein hormone? Yes, we gave you 3 examples of protein hormones. And those were insulin, a growth hormone, and oxytocin, and also glucagon. We've explicitly talked about those. You'll notice that there are genes that affect other types of neurological problems, like Tourette syndrome. So again, but what these are actually for, these genes, is instructions for proteins. And when there's a defect in the gene, the person cannot make a normal protein. It might be an enzyme, might be a hormone, might be a structural protein. You'll notice in the bottom row, and I'm just throwing these out, these are just examples of the roles of genes on our chromosomes and the kinds of proteins they code for. Here we see the genes that determine hair color and blood type. Again, I'm sorry, not blood type, but hair color and eye color. But again, these are really proteins. Interestingly, you'll notice that there is a gene here that also affects the cholesterol levels in the bloodstream. The cholesterol levels. So a defect in this gene can result in what's hyper? High, elevated, cholesterol, that's cholesterol, emia. What's emia mean? In the blood. Did we learn that last time? Right, we talked about hyperglycemia and ketonemia. Emia means in the blood. So if somebody inherits this defect in that gene, then they always have abnormally high cholesterol levels, even when they don't eat any foods with cholesterol, because there's a defect. If you don't have that problem with the gene, there's some people who luck out because of genetics. They can eat anything they want. They never develop high cholesterol levels. All right, and just to kind of wrap this up, you'll notice that we know that the sex chromosomes determine gender or sex. But the X-sex chromosome, which is much larger than the Y, has other additional genetic information on it, including genetic information that determines whether you can see in color or not, and also whether your blood can clot normally. Now, the Y chromosome doesn't have that information. So we had mentioned I think previously, that guys, where they have only one X chromosome and a Y, are much more likely to have the problems of color blindness or hemophilia. Females are less likely to have these problems because females have 2 X-sex chromosomes, not just 1. And in order for a female to have color blindness or hemophilia, they'd have to have defects in both of those X-sex chromosomes. But if a guy has a defect in just one of them, they've got the disease, because they only have 1 X chromosome. So the sex chromosomes can carry additional information other than just determining gender. All right, so we have just been reviewing genes. Let's go back to page B13. On page B13, so now this ought to make sense. Each gene, which is also known as an exon, or exon segment on a chromosome, specifies the amino acid sequence of a specific protein. That's really what the gene is for, is the instructions for building a protein, the amino acid sequence. Most genetic mutations, defects in the gene, are harmful because it causes the production of a defective protein. So the defect in the gene leads to a defective protein. These genetic defects are known as genetic or inborn diseases. Cystic fibrosis, hemophilia, Tay-Sachs, albinism, diabetes, even cancer, all have a genetic basis, which was really what we pointed out at the very beginning of the class, right? The genetic foundation to everything. That's why there's so much emphasis on genetics nowadays. Who's interested in pharmacy? So you're going to be hearing there's a new area related to pharmacy and pharmacology called pharmacogenetics. Has anybody ever heard that term? Because, you've heard it? Because depending upon somebody's genetic makeup, that may determine which medicine would be better for them to take versus a different medicine because of their differences in their genetics. That never used to be a factor because they really didn't analyze genetics. They gave everybody the same thing. But now we know people are genetically different and you don't give the same medicine to everybody because they react differently. Now we appreciate that better. So you will have a course in pharmacogenetics. Okay. Let's again talk about proteins very briefly. We'd already reviewed a little bit about proteins previously. Proteins, well, let's remind you of this. After water, which makes up about 60% of our body weight, what's the next major type of chemical that makes up our body? Proteins. So these proteins that are made based on our genetic makeup make up our whole body. They include structural proteins like collagen, which we've already been reminded. Collagen's found in our dermis of our skin and our tendons. It's found in our ligaments. It's found in our bones. It's found in the gums of our mouth, future dental hygienists. So it's very important. Keratin. Where's keratin found in the body? Okay, first of all, your nails are pure keratin protein. Your nails. Pure keratin protein. Your hair is also keratin, but it's got pigmentation as well. There's also some keratin in your skin. There are protein hormones, as we've learned. Insulin, glucagon, oxytocin, growth hormone are all examples of protein hormones. And there's a lot more. We're trying to expand our list of chemical names, because that's what physiology is about. There are proteins related to contraction. So you've all had anatomy, and you've learned about actin and myosin in muscle cells. Those are proteins involved in contraction. There are antibody proteins, which in a microclass are called immunoglobulins. And anytime a chemical has the ending globulin or globin, it means protein, like a globular-shaped protein, like a coiled up slinky. It's a globular shape. There are transport proteins.
You'd say, what do you mean?
Proteins that transport other chemicals. What's an example? Hemoglobin. First of all, it's a protein, it's a globin. What does it transport? Oxygen. Carries oxygen in a red blood cell. Some other transport proteins that we're going to be learning about are HDL and LDL. I'm sure you've heard of them. Anybody know what HDL stands for? All right. You know what, huh? Healthy? I don't know what healthy is. It stands for high density lipoprotein. High density lipoprotein and low density lipoprotein. Now a lipoprotein, or pronounced lipoprotein, is a protein that transports a fat. Right? These are transport proteins. This transports oxygen, these transport fats. Specifically, they transport cholesterol. Now some of you, we're going to learn more about this later in the course, some of you have heard this phrase, good cholesterol and bad cholesterol. Ever hear that? Cholesterol is cholesterol. There's no such thing as, well, you know, you got the good cholesterol and the bad cholesterol. No, what's good and bad are these lipoproteins. The HDL, the high-density lipoprotein, is the good one. You can think of the H for healthy. The LDL, the low-density lipoprotein, is the lethal one. It is really the amount of lipoproteins, whether it's the HDL or the LDL, that are the good and the bad. That's what's good and bad. Cholesterol is cholesterol. What makes LDL bad, that's the lethal one, is that this protein, it's a lipoprotein, low-density lipoprotein, it transports cholesterol to the walls of your arteries. So if you have a lot of it, it's excessively transporting excessive amounts of cholesterol to the walls of your artery. And what disease is that? When you see a buildup of fat and cholesterol on the inner walls of your arteries? Atherosclerosis. So that's the problem. Cholesterol is cholesterol. It has to do with the important thing, what makes it good or bad, is where is it being transported. We'll learn more about that. Those are transport proteins. There are others. Now, we mentioned several times now, arguably the most important role of proteins in any living thing, including us, are enzymes. Proteins that catalyze biochemical reactions. There are thousands and thousands of different chemical reactions that occur in the cells of our body, and each one is catalyzed by a different enzyme. Now, what are tumor suppressor genes? So a tumor suppressor gene is a gene that codes for a protein. And an example of this is, and we mentioned, p53 gene. I just told you it was an oncogene. We said, now what is the p53 gene for? The purpose of the p53 gene, remember all genes are instructions for making proteins. The p53 gene is an instruction for making a protein that inhibits cell division. It slows down cell division. It slows down mitosis. Now, what if somebody has a defect in that gene? Then they can't make this protein, and so what do you think happens to the rate of cell division? It keeps speeding up. And this increased rate of cell division results in a tumor that may even be cancers. So that's how a defect in this gene works. But first, most importantly, before we worry about the defect in the gene, we should need to understand what's the purpose of the gene, the protein. What is that protein that gene codes for? What's that protein for? What does it do? And in this case, it inhibits cell division. So in fact, we asked that question on the top of page B17. On B17, we asked what would happen if there was a mutation in the p53 gene, and the answer is you get colon cancer. And the BRCA gene similarly increased risk of breast cancer. Okay, now the next thing, now that we've kind of given you an overview of what genes are and the proteins that the genes code for, we want to talk about DNA replication. To make a replica is to make a copy. DNA replication, making copies of the DNA, occurs in cells before they can divide into two cells, before they can undergo cell division, before they can undergo mitosis. Now, before I start to read what's written here, let's go back to page B14. Let's go back to page B14. Okay, so on page B14, it shows a cell. And this cell, they've shown, has four, technically the best term would be chromatids. But I'll use the term chromosomes, 4 chromosomes. And we know that our body cells don't have 4, they have 46, 23 pairs. All right? You'd say, well, then why didn't you show us a picture with 46 of them? Because it'd be a real mess to have a picture with 46 chromosomes. So to keep it simple, they just drew 4. Now, notice how these chromosomes duplicate. Can you see that? Where there used to be 1, 2, 3, 4, now each one has an additional mate, as it were. Now, why does that DNA replication occur? That has to occur before this cell can divide and become 2 cells. Because if you don't duplicate the chromosomes, if you don't make a copy of them, then when the cell becomes 2 cells, each cell will have only half as many chromosomes as the original cell. So if we're going to maintain that number of 46 chromosomes in every body cell, we have to, before the cell divides, duplicate or replicate that DNA. And then you'll remember from biology and from anatomy and all these other courses, they line up in the middle of the cell during metaphase of cell division, and then they pull apart, and we get 2 genetically identical daughter cells that are not only identical to each other, but identical to the original cell from which they... Let's see, how does the DNA, how do the chromatids or chromosomes, how do they duplicate? Now again, I'm not going to go into as much detail as you probably did in some of your more advanced biology classes. You'll recall that the DNA is really a double helix. We reviewed the structure of DNA back in the end of section 8. Right? We saw that it was like a twisted ladder shape. And that twisted ladder shape can either be kind of in the uncoiled chromatin appearance or coiled up chromosome shape. So here's our double helix, our twisted ladder. And there is an enzyme called helicase that basically causes the two strands of this twisted ladder to separate. And why it's called helicase is, remember, another name for this twisted ladder shape is a double helix. And helicase, the fact that it ends in ase, is an enzyme, so it causes the two strands to unzip. That's commonly the term we use, it unzips. Now, what happens, these are the two original strands, and so a new complement forms opposite each of the two original strands. When you learn this in biology, they'll commonly call this the semi-conservative theory of DNA replication. Because it means that when you look at now where we now have two double helixes where there used to be one, two DNAs where there used to be one strand of each one is part of the original, and the other strand is newly synthesized. So that's called, you don't have to know this, but semi-conservative model of DNA replication. All right, so this is how the DNA replicates. So when we draw a picture here, when we have a picture here, where we go from 1 to 2, this is what's occurring at the molecular level. At the molecular level. So here's how I summarized it. On page B17, on B17, where are we here? DNA replication. All right, it has to occur before a cell can divide. You actually learn about this in biology and anatomy. You learn that this DNA replication occurs during the S period of interphase. Remember the G1 growth, first growth period, the S period followed by the G2 period, and then the cell divides. So we wrote that the DNA unzips. It's really the enzyme helicase that does that. It unzips in the middle so that each strand of the original double helix acts as a pattern. And the term that the books commonly use is template. It acts as a template or pattern to reconstruct a new complementary strand for its half. Now, in order to build this complementary strand, you need another enzyme. It's called DNA polymerase. Helicase simply caused the original strand to unzip, but to build a new complement, you need an enzyme DNA polymerase. We actually mentioned that previously. In fact, we mentioned that it needs a coenzyme to work, zinc. So if you look back to your notes, we mentioned when we were talking about which vitamins and minerals you need, as coenzymes, we mentioned zinc. Right? There's somebody checking. That was near the very end of section A. And zinc is a coenzyme for DNA polymerase. So if you have the original DNA, you add this enzyme, DNA polymerase, and you need that zinc. And of course you need DNA nucleotides to build that complementary strand. You'd say, well, what do you mean DNA nucleotides? Adenines and guanines and cytosines and thymines, right? HECTs. And of course you need some ATP to build this, to make it, join it all together. Remember, when you break things apart, they release energy. But to build them, it takes energy. It's like, it doesn't take any energy to destroy something. It takes energy to make it, to build it. So you need some ATP. And basically, if you add a test tube, and you put some DNA in it, you put this enzyme in it, you add some DNA nucleotides, A's and G's and C's and T's, and some ATP, and stir it, you get multiple copies of that DNA. That's actually known as a DNA, a PCR reaction, a DNA polymerase chain reaction, a PCR reaction in biochemistry. All right, so that's how we make more DNA, and what's important is, why does it have to happen? It has to happen before one cell can divide into two cells so that we maintain the right number of chromosomes in each cell.